Hello, everyone, and welcome to today's webinar, Solutions for Myeloid Cell Research from Periphery to Brain. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Nizar Yousif, Global Product Manager, Immunology and Immuno-Oncology at Miltenyi Biotec, and Dr. Elisa Nent, Global Product Manager for Neuroscience at Miltenyi Biotec. Nizar and Elisa, you may now begin your presentation.
Thank you for the kind introduction, and hello everyone from my side. Today, my colleague Elisa and I are going to take you on a journey through our solutions for myeloid cell research. I will cover the first part and we'll be talking about solutions for myeloid cells in their periphery. Elisa will then continue the presentation and talk about solutions for myeloid cells in the brain. We hope that you enjoy this webinar. This is our agenda. I will start with a brief introduction about myeloid cells. Then we'll talk about our sample preparation solutions for myeloid cells in general. Afterwards, we go through the solutions for different myeloid cell types. And today, due to the sake of time, I will focus on solutions for monocytes, macrophages, and neutrophils, while Elisa will focus on solutions for brain macrophages, namely microglia. Myeloid cells are diverse and beautiful. They possess different shapes and size, as you observe here in this slide. Myeloid cells are present everywhere. You find in the liver, Kupffer cells and dendritic cells. If you look at the spleen, you find red pulp macrophages, dendritic cells, as well as granulocytes. In the lung, alveolar macrophages and also dendritic cells. Present in the blood are granulocytes, monocytes, and dendritic cells. And an example of myeloid cells in the bones are osteoclasts. In the peritoneum, we have peritoneal macrophages and dendritic cells. In the skin, we have the famous Langerhans cells and the skin macrophages. And in the most important organ in the body, the brain, we have tissue-resident macrophages, specifically named microglia, and also perivascular macrophages. And in tumors, we have tumor-associated macrophages, TAMs, tumor-associated neutrophils, TANs, and myeloid-derived suppressor cells, MDSCs. So please note that these are examples and not restrictions. The widespread presence of myeloid cells in different organs immediately gives us the indication that these cells have important roles. So let's have a look together at some examples of myeloid cells functions. Myeloid cells have important functions in health and disease. For example, in healthy state, tissue macrophages are highly specialized in removal of dead cells and cellular debris and thus maintain proper organ function. Moreover, macrophages are involved in metabolism of iron, calcium, lipid, and other substances and contribute to the maintenance of these substances in the body. In case of infections, macrophages engulf pathogens and destroy them, while neutrophils are highly motile and quickly react to chemical signals such as cytokines secreted by other immune cells like macrophages during infections and migrate to the site of infections. Neutrophils have cool methods to attract and kill pathogens, including the formation of neutrophil extracellular traps, as you observe here in this image. Neutrophil can also do phagocytosis to destroy pathogens, or they can release antimicrobial agents from their granules to destroy invading pathogens. In case of cancer, myeloid cells can have positive or negative outcomes. They can contribute to tumor progression by suppressing anti-tumor immune cells. Here I'm referring to myeloid-derived suppressor cells, MDSCs. And research has also shown that macrophages may have cytotoxic activity to kill tumor cells directly. And myeloid cells have also been shown to affect cancer therapy in positive and negative way. Now, let's talk about the sample preparation solutions for myeloid cell research. So, careful preparation leads to a happy end. And here I'm going to show solutions for human and mouse myeloid cells. So, what are the challenges for myeloid cell sample preparation? Well, time is an important factor. Some researchers process several samples, which requires long preparation time. Having low number of viable cells is a big challenge in many experiments. And also epitope degradation due to improper sample preparation is a problem for downstream applications, for example, flow analysis or functional analysis, where the cell surface receptors are needed. Having inconsistent yield is a challenge for reproducible results. So what is the solutions, or what are the solutions? As the solutions for these challenges, I'll introduce you our GentleMax technology and associated tools. The GentleMax dissociator is a benchtop instrument for semi-automated dissociation of tissues into single-cell suspensions or thorough homogenates. We have two types of unique GentleMax tubes used with the instrument, and these enable the time-saving and easy dissociation or homogenization of tissues in a closed system. So the combination of mechanical disruption using the GentleMax and its different tubing systems with the enzymatic digestion using optimized and highly purified enzymes in this technology enables efficient dissociations. So speed, viability, epitope preservation, and reproducibility are guaranteed. Now, I would like to show you flow cytometry data generated after dissociation of different mouse tissues. Single-cell suspensions of spleen, lung, lamina propria, and tumor tissues were obtained with the GentleMax and the associated dissociation kit. As you observe here, this enabled obtaining of high viable cells, which allowed the detection of disease subsets. So cell surface epitopes were well preserved for labeling with antibodies against relevant phenotypes markers. Now, I will carry on and talk about solutions for monocytes and macrophages. Here, I will cover solutions for human and mouse monocytes and macrophages. And we start with the solutions for human samples. Here we have a wide range of solutions for sample obtained from different materials, from peripheral blood, whole blood, buffy cord, LRHC, leukopacs, or PBMCs. Solutions for monocytes and macrophages separation, shown here in blue. Cell culture solutions for monocyte differentiation into macrophages, solution for macrophage polarizations. This includes cytokines and growth factors, shown here in red. Different antibodies for the analysis of these cells, shown here in green. Now, let's start with some of the cell separation challenges. Cell sorting requires long time, long number of viable cells, which affects various downstream applications, and a specific cell activation. You don't want to have that on your separated cells for reliable downstream applications. Having few cells of interest after separation is a challenge for conducting your experiments. Now, let's talk about our cell separation solutions for these challenges. We offer several solutions for the isolation of monocyte subsets for different starting materials. Solutions for pan-monocytes or isolation of pan-monocytes. We have variable options for the isolation of classical monocytes and a solution for the isolation of non-classical monocytes. In this slide, you observe the isolation of monocytes from human PBMCs using CD14 macrobeads, which is a positive selection method. Positive selection means that during separation, monocytes are magnetically labeled and remain retained within the column, while unlabeled cells flow through. After a washing step, the column is removed from the magnetic field of the separator and target cells are eluted from the column. As you observe in this flow analysis results shown here, high purity and high recovery of the isolated CD14 positive cells have been obtained. Another option for monocyte isolation is by using the pan-monocyte isolation kit. The pan-monocyte isolation kit is an indirect isolation method for the isolation of untouched monocytes from human PBMCs. It allows the enrichment of classical, non-classical, and intermediate monocytes. So by using this kit, you get your highly pure monocytes by the depletion of the magnetically labeled non-target cells. The electron microscopy results you observe here is from a recent publication in which they showed that monocytes isolated from COVID-19 patients have dysfunctional mitochondria. In this work, monocytes were isolated using our pan-monocyte isolation kit. And as you observe here, no monocyte dysfunctions have been detected in the control. Before I introduce you to our straight from solutions for monocytes, let me say some few words about our straight from technology. A straight from technology enables the separation of highly pure cells directly from different blood sources with all the benefits of max microbeads, as well as easy and fast enrichment of leukocytes. So no need for density gradient, no cell counting, no washing, and no erythrocyte lysis needed. And this is an example of multiple separations from one sample using straight from technology. This technology permits multiple cell isolation of CD14 positive monocytes, CD8 positive T cells, and CD4 positive T cells from the same sample in just 1 hour time. And as you observe here, we have several straight from technologies for the isolation of monocytes starting from different materials. A fitting solution I would like to introduce here for line sample processing is cell separation via automation. The Multimax Cells 24 separator nicely fits for this purpose. It has multiple practical features, from flexible sample scalability to processing of large sample volume in short time. Now, an alternative option for the isolation of human and mouse monocytes and macrophages is via the CD14 macrobeads. These macrobeads are developed for the isolation or depletion of human and mouse cells based on their CD11B expression. And in these plots, you observe CD11 positive signal of mouse splenocyte before separation, a splenocyte without CD11 positive cells. These are cells in the flow through and the nicely isolated CD11B positive mouse cells here on the very right. Next, CD163 microbead kit human. This kit allows the enrichment of human cells from, for example, healthy or tumor tissues based on the expression of the CD163 antigen. CD163 is most abundantly expressed by mature tissue macrophages and peripheral blood monocytes. You observed here result from CD163 positive cells isolated from ovarian carcinoma sample using the CD163 microbead kit. After isolation, the cells were stained with CD163PE and CD206 Fitch antibodies are analyzed by flow cytometry. The isolated cells are ready to go for different downstream applications. Now, let's move to the solutions for mouse monocytes and macrophages. And here we also offer several options for different sample sources from cell separation to cell culture to cell analysis. And these are solutions for myeloid cells in the lung. Now, anti-F480 macrobeads ultra-pure is very good option for the isolation of mouse macrophages from spleen or tumor tissue. And here below is flow data showing the separation of F480 positive cells from spleen and tumor tissue. In this slide, I would like to show you that macrophage is isolated by anti-F480 macrobeads ultra-pure are ideal for downstream molecular analysis. Macrophage is isolated from tumors from breast cancer xenograft using anti-F480 microbead ultra-pure were analyzed for innate immunity pathways using the NanoString platform. The general message of this result is that treatment with different drugs modulated macrophages gene signature. For the isolation of mouse monocytes and macrophages from blood or lymphoid tissue, we have the CD11b macrobeads ultra-pure. You observe here in the dot plots CD11b positive cells isolated from spleen using CD11b macrobeads ultra-pure mouse, stained with CD11b Fitch and anti-GR1p antibodies and analyzed by flow cytometry. On the first dot plot, the cells shows the cells before separation. In the second dot plot shows the flow through without the CD11b positive cells that are returned in the column, and the third dot plot shows the isolated CD11b positive cells. So to wrap up the section of monocytes and macrophages, our solutions, cell separation solutions for these cells guarantee time saving, optimal viability and functionality, as well as reproducibility. And let's start with solutions for neutrophils from human models. Here you observe our options for human neutrophils research. The challenges I mentioned in my previous slides for other myeloid cell types exist here as well. So I don't need to go through it in details again. So what are the solutions for these challenges in case of human neutrophils? Well, let's have a look together at our Max Express technology. Human neutrophils are usually obtained by density gradient centrifugation. However, this method is exceedingly time-consuming and frequently results in neutrophil populations that are contaminated by other leukocytes, mainly eosinophils. With the MaxPress technology, untouched human neutrophils can be isolated from whole blood within 20 minutes. And in this method, erythrocytes are aggregated and sedimented, and non-target cells are removed by immunomagnetic depletion with the Max Express beads, and the untouched, highly pure target cells can then be easily collected, as you observe here in this illustration. In these results, you observe that human neutrophils are efficiently isolated from whole blood using Max Express neutrophil isolation kit. In the upper dot plots, you observe the cells before separation, and in the lower dot plot, you observe the cells after separations. Here, after separation, the cells were stained with CD15 and CD16 antibodies, which are markers for human neutrophils. This kit allows isolation of neutrophils with high purity recovery, and viability in a short time period. And here are all the nice features listed. Here, the functionality of neutrophils isolated with Max Express technology was tested. The neutrophil isolated with Max technology show high ROS production upon stimulation with E. coli, red bar, and PMA light blue bar, as compared to the neutrophils isolated with the density gradient method. This data was obtained from flow cytometry analysis. In this slide, I would like to show you data from a publication where it was assessed whether oxidative metabolism might be present in neutrophils from cancer patients. In this work, they isolated neutrophils from peripheral blood using Max Express neutrophil isolation kit. And analysis revealed that patients harboring ovarian cancers had an increased proportion of neutrophils with intermediate expression of CD10 and CD16, which is characteristics previously reported as indicative of immature phenotype. And immature CD10 intermediate neutrophils possess greater mitochondrial content. And cancer patients' neutrophils possess greater ATP synthase dependent OCR. OCR states for oxygen consumption rate compared to healthy controls. Now, let's go to the solutions for mouse neutrophils. Here we also have rich solutions for different tissues. Now, the anti-LY6G microbeads ultra pure for the isolation of mouse neutrophils from lung tissue, blood or bone marrow. And in these top plots, you observe the cells before separation and the effectively isolated neutrophils as indicated by CD11B and anti-LY6G staining. The neutrophils isolation kit is an alternative option to the anti-LY6G microbeads ultra pure if you are interested in obtaining untouched mouse neutrophils, since in this kit the untarget cells are magnetically depleted and the target cells are isolated without magnetic labeling. Again, here the cells before isolation and the isolated neutrophils are shown. Now, these are results from a very recent publication where they conducted transcription analysis of neutrophils isolated from mouse liver and blood. The dot plots are representative flow cytometry profiles of isolated neutrophils. On the left, forward versus side scatter plots. On the right, LY6G versus CD11B staining profiles of the neutrophils. The graphs on the right show gene expression analysis on mouse neutrophils. So I'm getting close to the end of my presentation, and in my last slides, I will talk about our cell analysis solutions for myeloid cells. So once you get your cells of interest, of course, you want to analyze them. Here in the cell analysis part, we also have several challenges. One of the challenges could be that the antibodies are not highly specific, so they are not effective in capturing the antigen of interest. Moreover, having high background signal where antibodies detect signals that are not specific to the antigen. One of the reasons for high background signals is non-specific binding of antibodies to Fc receptors found on myeloid cells such as macrophages, monocytes, and dendritic cells. So let's have a look at our solutions for this part. You'll be able to do precise analysis of your myeloid cells with our reaffinity antibodies. These are recombinantly engineered antibodies, so lot-to-lot consistent. They are highly pure, and they consist of one universal isotype control, so one human IgG1. And most importantly, these antibodies are engineered to lack Fc part, which lead to prevention of unspecific binding. This is an example of human macrophages phenotype using reaffinity antibodies. The results show that CD163, CD209, and CD200R are highly expressed in M2 macrophages, while CD68 is highly expressed in M1 macrophage. The complete story, including solutions for macrophage polarization, cell culture, and phenotyping, is included in an app note, application note. I inserted a link for it here. So for more details about this topic, please have a look at it. Now, I'll wrap up the first part of this webinar, and here are some take-home messages. The GentleMax dissociators and the dissociation kits are here for optimal myeloid cell sample preparation. We have optimized separation of myeloid cells, so positive isolation, untouched isolation, or straight from automation. Multimax, Automax are mostly possible with our cell separation options. The cells isolating with our cell separation technology are suitable for many downstream analysis, omics, cell culture, flow phenotyping, etc., etc., and precise analysis of myeloid cells with reaffinity antibody, so consistency, standardization, and high specific signals. And most importantly, our experts are happy to guide you through the entire workflow for myeloid cell research. And here, I will hand over to my colleague, Elisa, and I thank you for your attention.
Okay, thank you, Nizar, and welcome also from my side. I will present to you now the solutions for brain myeloid cells. So this here is the human brain, and as most of you know, these here are the main neural cell types. We have neurons, astrocytes, and oligodendrocytes, and there are actually two more types of cells in the brain. These are the microglia and macrophages. And although microglia are also called tissue-resident macrophage, they are actually quite different. So you can see here that these cells reside in different niches in the brain. Microglia are located in the brain parenchyma, whereas classical macrophages can be rather found close to vessels, at the choroid plexus, or in the meninges. Each cell type expresses a unique set of markers and has slightly different function. So for now, let's focus on the microglia. In general, they have several functions, as you can see here. In the healthy brain, microglia support neuronal development and survival, promote synapse formation and remodeling, modulate neuronal activity, or phagocytose dead cells and myelin. Whereas in the case of a tissue damage or neuroinflammation, a microglia can get quickly activated and phagocytose synapses, which may lead to neurodegeneration, or can activate astrocytes by releasing pro-inflammatory cytokines. So now the big question is, what's the best way to study microglia cells? If you've worked with microglia before, you maybe know that isolation can be sometimes tricky. You can either sort those cells or have mixed clear culture. But actually both approaches need a rather harsh tissue homogenization, which can stress the cells. And moreover, cultivation takes around 2 weeks, especially if you need high cell numbers. Additionally, if you've ever dissociated the adult brain, you maybe encountered some problems due to the high amount of debris and blood vessels. And to tackle all of these problems, let me introduce you now to our workflows for neural cell analysis. So as you can see here, we have a dedicated workflow when analyzing the brain. It goes from sample preparation over to cell separation and can be followed by cell cultivation or analysis. Depending on the age of the animal, we recommend a neural tissue dissociation kit for neonatal samples, or our adult dissociation kit for the adult brain. We have more products for cell isolation and analysis that I will present to you further in the following slides. And additionally, you also have the option to follow our imaging workflow that allows you to clear whole brains or hemispheres and image these organs in 3D with our ultramicroscope lathes. But for today, I want to focus on our cellular workflow. So let's start with sample preparation in the first step. The GentleMax octodissociator with heaters helps you with a standardized and automated dissociation. It uses mechanical disruption and enzymatic digestion and thereby performs a gentle and efficient brain dissociation. And as I mentioned earlier, adult brain tissue is a bit tricky to work with because it can generate huge amounts of cell debris and red blood cells, which then hampers downstream applications such as cell isolation, cell culture, or single-cell sequencing. And our adult brain dissociation kit works together with the GentleMax octodissociator with heaters and includes a step for debris removal and red blood cell lysis. This then helps to remove the debris efficiently and also removes the red blood cells without the need of performing animal perfusion. As you can see also here in the figure, it results then in a high yield of pure neural cells. After the sample cleaning, you are able to isolate the microglia with our CD11B microbeads. This magnetic cell separation technology is a gentle and fast way to obtain your cells of interest from the cell suspension. It takes less than an hour and it will result in a viability of around 95% and a purity of up to 98%. I guess most of you have heard of this product already, since Miltenyi is famous for the MAX technology, and actually the CD11B microbeads are also well known in the field. Then cells isolated from our workflow are highly viable and can be even cultured afterwards. So for this, we offer a serum-free cell culture medium and optimized supplements to cultivate not only microclear, but also neurons, astrocytes, or oligodendrocytes. This can be especially useful if you are interested in a co-culture of several cell types. The serum-free formula prevents the high variation between the experiments that can normally result from using different FCS lots. And to boost your microclear culture, we offer high-quality cytokines like the MCSF that will increase your cell culture yield greatly. If you are working with PSC-derived neural cells, we also have the optimized STEM-Max PSC Xeno-free media that allows for a weekend-free feeding schedule. So we also offer various antibodies for neural cell applications, which are suitable for flow as well as for immunofluorescence. Some of them can be used with a very fast protocol and only need around 10 minutes incubation time. So this is then actually quite nice and can save you also some time in the lab. Additionally, we are also in the process of switching our hybridomers to recombinantly engineered antibodies, which will result then in a higher lot-to-lot consistency and less background staining. And now I would like to show you some highlighted publications that may help you develop your own protocols. So the first one was from a customer which was published last year as a STAR protocol by the lab of Stacy Bilbo. They describe step by step how to isolate viable microclear with our CD11B microbeads from the adult mouse or even the human brain. And for anyone that is interested in this method or is working with human tissue, I can greatly recommend this paper. But if you're more into PSC-derived cells, I would like to present to you this publication. The study was published beginning of this year by Renzo Mancuso and his group. They developed a new method to differentiate human PSCs into microclear and then transplant them into the mouse brain. This way, they are able to study human microglia cells in their normal environment. For these analyses, they used our adult brain association kit, our antibodies, and most importantly, the Max Planck Institute cell sorter, which is an instrument for very gentle cell sorting that is based on microchip technology. And in the last paper, I want to introduce you to the ultramicroscope Blaze that I've also mentioned before in my previous slides. This fully automated light sheet microscope can image large or multiple samples using cutting-edge optics. In this publication here, they use the ultramicroscope to study the cleared mouse brain in Alzheimer's disease, to quantify plaques and thereby help to find new approaches to treat this disease. And before we come to an end of this presentation, I would also like to mention shortly our specific products for the inflamed brain. For several disease models, it is beneficial to also analyze infiltrating immune cells in addition to the neural cells. And for this, we offer the multi-tissue dissociation kit and a set of different immune cell microbeads. In the case of EAE, which is experimental autoimmune encephalomyelitis, which is a model for multiple sclerosis, this workflow can then easily help to isolate and analyze neural tissue. So here you see an example of isolated immune cells from a brain of a EAE mouse with our inflamed brain workflow. You can clearly appreciate the different cell types that are present in the inflamed brain after the blood-brain barrier got leaky and immune cells were able to infiltrate the brain. And this workflow can be even used for the analysis of immune cells from small tissues like the spinal cord, as you can see here in these plots. And with this, I will come to a summary of my second part. I hope I could show you that the adult brain association kit and the GentleMax octo-dissociated with heaters allow for standardized, gentle, and efficient adult brain association, which results in a highly viable single-cell suspension that is free of debris and red blood cells and that is suitable for different downstream applications. The Max microbeads and the Max column technology enables an easy and fast isolation of pure, viable, and functional microglia. The Max Quantito cell sorter allows for microchip-based cell sorting in a fully closed and sterile cartridge system. This gentle method is especially suited for sorting of fragile neural cells like microclear. And last, with the ultramicroscope Blaze, you are able to image large or multiple samples in an easy and automated way. 3D imaging of the cleared brain allows you to visualize the location and connection between the different neural cells. And with this, I'm at the end. I want to thank you for your attention. I hope I could show you our dedicated solutions for your myeloid research. And now please don't hesitate to ask if there are still open questions, and for sure you can also always contact us afterwards.
Thank you, Nizar and Elisa, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, I would like to isolate human neutrophils from whole blood, both normal density and low density neutrophils. Would it be possible to do that with the Express kit you presented?
Well, with the Max Express whole blood neutrophil isolation kit, human are not, neutrophils are not isolated based on density. So therefore, both normal density neutrophils and low density neutrophils should be isolated. So, and in the Max Express technology, the aggregated erythrocytes are sedimented at the bottom of the tube. So whereas non-target cells are magnetically labeled and adhere to the tube walls as long as the tube is placed in the magnet. So I don't see any difficulties for getting your neutrophils of interest using this kit. Yes.
Thank you. Next question. I am trying to isolate infiltrating monocytes from inflammatory adult rat brain from the hippocampus. What is the best option? What is the best antigen to distinguish them from microglia?
Okay, I think this I can answer this or try to answer. Yeah, it's a bit tricky to distinguish monocytes from microglia, right? Since they're quite similar. And we don't have microbeads for rat monocytes, so it wouldn't work with microbeads and magnetic cell isolation. But what I would suggest is to use, for example, the Taito cell sorter. So you could sort your cells and you could use different kind of markers. So either you go for CD11b, CD45 high. You could also go for Ly6C high or CCR2 high. And if you want to be sure to kind of omit all microclear, you could go for TMEM 119 low, because this marker is quite unique for microclear. And if this marker is low, it should be a monocyte. So you could try those options for sorting. But I think magnetic cell isolation wouldn't work in this case because those two cell types are too similar in case of the inflamed brain. I hope this helped you a bit, but for sure you can also write me and I can also try to send you additional material to those products.
Great, thank you. Next question. Are there differences in the downstream applications of the different monocytes isolation kits or microbeads, for example, for the differentiation of monocytes into macrophages?
Yeah, I'll go for that. Yeah, no, not at all. Yeah, so using either positive isolation of monocytes with, for example, CD14 macrobeads or untouched isolation with monocyte isolation kits, you will be able to differentiate the isolated monocytes in culture into macrophages. Of course, with the help of specific cytokines and stimulation factors. Yeah, it's just a matter of preference. Yeah. Some researchers have long experience with positive isolation and prefer to stay with it, and others are used to the untouched isolation methods. Moreover, there are good literature available. Yeah, so where researchers are using our different monocyte isolation methods to isolate monocytes and differentiate them into macrophages. Yeah, so this is, you can have a look at it in PubMed. Yeah.
Great, thank you. Next question. You presented straight from products for the isolation of cells from human blood sources. Do you have straight from solutions for the isolation of cells from mouse tumor tissue?
Yeah, this is actually very, very interesting. Yeah, very nice question. Yeah, no, we don't have straight from solutions for mouse tumor tissues yet. Yeah, so however, we are working on mouse tissue straight from solutions, for example, for the isolation of myeloid cells from mouse spleen. So please stay tuned.
Great, thank you. Next question. Any solution for microglia cells counts? Do you have any solution for that?
Yeah, thanks for the question. I'm not 100% sure what is meant, so maybe you can also elaborate a bit on this question. But basically, if you use the CD11b beads for the isolation of microglia, you get quite high numbers. If you are kind of need like a high cell count or high number of microglia, and also then of course it depends on what brain region you use, and basically then also on the age of the animal. But please, if you can put another question and explain a bit, and then I can try to answer.
Great, thank you. While we're waiting for that, our next question is, can you elaborate on the activation status of monocytes after magnetic isolation?
So actually, yeah, this question is quite common. Yeah, we get it so often. Well, our internal data, some of these data is available online in our Select the Best brochure. Yeah, so this data confirmed that monocyte isolation with the Max technology does not lead to the activation of these cells. Yeah. So the advantage of our Max technology is the minimal labeling, so maximum 30% of the surface epitopes will be bound by antibodies. And also the microbeads are tiny, so therefore they do not lead to cross-linking or triggering of surface receptors, and thus the activation of these cells. Yeah. So, and moreover, this is also proved by publications, for example, where monocyte isolated using Max technology from healthy controls did not show activation of inflammatory markers.
Thank you. Next question. Why is it so difficult to isolate cells from the adult brain?
As I tried to explain a bit in my slides, basically the older the mouse gets, or the older the brain is, the more vascularized and more red blood cells are there. And also you have more neurons. This means more myelination around the neurons. And in the end, you end up with more myelin debris and more red blood cells. And this can hamper your downstream applications. So either if you would do a directly flow analysis, you would really get a lot of events, and it might be very difficult to analyze your cells of interest. It wouldn't be really pure. Or if you use microbead isolation, then it could also block the binding, and then you would also not have a great result or a high yield. So that's why this adult brain association kit that we developed is specifically for the adult brain, and it tackles those two problems. So you don't have to worry about those two debris and red blood cells, and you get a quite pure and high cell yield in the end.
Great, thank you. Next question. What are potential other downstream applications of the isolated cells?
So in case of microglia, so it's after the microglia cells, after you isolate them from the brain, I mean, what I showed mainly were the cell culturing, right? But you can, of course, also go for other applications, such as single cell sequencing. So we made sure to collaborate with 10x Genomics and really check that our workflow is compatible with their single cell sequencing, so that you can, or you have the option also with this established workflows of ours to further analyze your cells and then do a single cell sequencing, for example. So this would be also another option that I really haven't mentioned much yet, but it's also possible.
Thank you. Next question. How is the Max Quant Tito cell sorter used for the analysis of PSC-derived microglia?
So this was also something that I didn't go so much into detail. Basically, the Tito was used in this case to sort the microglia, especially before transplantation. And the good thing about this is the Tito uses a system like a cartridge system, which makes the whole process sterile. So this was then really helpful in this specific paper or protocol, because Renzo also needed to transplant cells back into immunodeprived mice. So of course for this they had to be quite sterile, not to cause any further inflammation. Exactly, and that's how the Tito was really helpful for his study of the PSC-derived microglia.
Thank you. Next question. Can you elaborate a bit on the imaging workflow?
Yeah, sure. So the imaging workflow, I also didn't really show it. I mean, I just showed it shortly in the beginning. Basically, it's you can image and analyze hemispheres or the whole brain. And the workflow would be that you start by staining the organ with your antibodies of choice, and we are constantly increasing our set of antibodies that are especially made for 3D analysis. Next step would be the clearing. So for this, we also have a clearing kit, which is quite easy to follow, and you don't have to go through the thousands of protocols that are available on the internet. And then obviously in the last step, you would image the brain or the hemisphere. And with this ultra microscope blades that are presented, it's really nice. You can image several samples in one row. It's quite automated. So you can have like a batch analysis and imaging. And then in the end, you get really nice 3D videos or images of your brain, and it's really helpful for the connectivity or the interplay of the cells, or for example, also during development.
Thank you. Next question. Why do you have to use a different dissociation kit for the inflamed brain?
For the inflamed brain, so what I showed you here, we recommend the multi-tissue dissociation kit because if you are working with the inflamed brain and you are interested in immune cells, so in infiltrating immune cells, you need to make sure, or at least if you want to isolate those immune cells later on with our microbeads, you need to make sure that the kit and the beads fit to each other. In case of the neural cells and the adult brain association kit, this works. But for some of our immune cells, the epitopes are quite sensitive. So the multi-tissue dissociation kit works better. So this is why depending a bit on the cell type you're interested in, we recommend different kits or slightly different adapted workflows. So the best would be always, if you have a really specific question in mind, to write to us or write to our tech support, then we can try to set up with you the perfect protocol or what products you need or what would be suited the best.
Thank you. Next question. Is it possible to isolate microglia from P8 mice of the hippocampus region?
Yeah, I think it's possible. So we have this, not only the adult brain association kit, but also for younger mice, the neural tissue dissociation kit. P8 is kind of in the middle, let's say. But since I guess what you need would be just like a small region like the hippocampus, right? And since you have basically less amount of tissue, the workflow or the protocol of the adult brain association kit might be a bit tricky. So here we would recommend a neural tissue dissociation kit because at P8 we don't expect so huge amount of debris or red blood cells, so you don't really need to do those steps. But you can still isolate them with the beads, so this should be perfectly fine also from a smaller brain region. And I think we even have protocols available online to do this specifically for small brain regions. I can also send you some if you want later on. But yes, this is possible.
Well, thank you. It looks like we are out of time for questions. Thank you, Nizar and Elisa. Do you have any final comments for our audience?
Yes, so I just would like to thank you very much for joining our webinar today. And if you are looking for detailed information regarding myeloid cell research solutions, please feel free to contact us per email or visit Miltenyi Biotec website. And I hope to have you around again in our future webinars, and have a nice day or evening wherever you are. Yep.
Thank you also from my side.
Well, thank you again, Nizar and Elisa, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.